second. I think we will start, and when he comes, he will join. Okay. So today we will discuss motor learning and skills. And I think I will give quite big introduction. And then, wait a second. Yeah. And then we will continue with a short paper. Andrei will come and present a paper at some point, I think, but introduction will be long, so he will have enough time. And I will discuss not only behavioral approaches to study motor learning and skills, and also about a little bit about anatomy and introduction, not so deep, but in case somebody forgot how it works, because when I was preparing, I was like, I don't really remember how motor skill works, like cerebellum, yeah, whatever. So I think it's quite interesting. So let's try. If you have any question, you can interrupt me along the way, as usual. Okay. So I don't like how it works. Maybe like this. Oh, okay. I think I did something wrong. Wait. Oh my God, I'm retarded. But can you see something or not? No? Ah, yeah, you can? Okay, because I didn't see myself. Okay, well, it should be like this, my screen. For some reason, I don't see you when I gallery, multi-speaker.
We do see the understanding skill memory slide though.
Okay. Well, I will look at myself then. I will not see your faces. So, shit things happened. Then let's try to understand what is a skill memory. Skill memory, it's knowledge about how to do something. It differs from episodic or semantic memory when we remember something like what happened, or like what event was that, or like the earth is round. We just know how to do stuff. We know how to brush our teeth and whatever. And there is a difference between memory about what and how, because skill memory have specific qualities. It's usually long lasting. So for example, if you manage to learn how to ride a bike, even with a break, you can recap that skill really fast. But sometimes with memories about what, it's of course when you relearn something, it's kind of faster, but with skill memory, it requires less effort to recap and to start doing something again. Or sometimes you even don't need extra practice to do something after a break. And also, it's non-declarative, so it's non-verbal. Riding a bike does not require any verbalization because yeah, with episodic memories, we also can say, like, if I remember some episodes from my childhood, I also have a visual representation of that. But usually, it's especially with semantic memories, it's more descriptive, and you should put it into words. When we study like neuroscience or something, we usually put it into words. But yeah, hello. But with skill memory, we don't need that. And we can divide skill memory in two main branches. It's cognitive skills and motor skills, because there are some cognitive skills that we also can master, like solving some type of puzzles, and then we can do it more or less automatic. Yep. We just started, so you didn't.
Sorry for being late recording stuff.
Yeah. So let's talk a little bit about acquiring skills. We discussed during first lecture Edward Thorndike. I don't like his surname. It's inconvenient. So it was the guy who invented puzzle box for cats. He worked on not only on cats but also on humans. And what he did, he asked humans to draw a line about 3 inches long. I don't know how many centimeters inches are, but it doesn't matter. So in this experiment, he divided people in 2 groups. One group received some feedback and another group didn't. So a group that received feedback on their actions, like person draw a line and then he received immediate feedback like, yeah, this time it was too long or too short or something. They mastered skill way faster than people who just trained to do 3 inches long lines and then received no feedback. It doesn't make any sense. So by that, he highlighted that feedback is really important for acquiring skills. Yeah. And also, people studied the dynamics of skill acquiring, like how it works. And so at the beginning, performance increases really fast, but then it's going to plateau. Like here in a reading task, participants acquired skills really fast at the beginning, but then like they had to read a page and the speed of the reading the page was kind of estimated. And then at some point it's plateauing because it's difficult to improve more and more. And in order, it's like with English, I don't know, at the beginning you jumping to B2 or something really quick, but then in order to like from C1 to go to C2, I never did. So it's really difficult and slow. And at least in motor learning, like in dancing or something, people show that observational learning really can help to overcome that plateauing effect. And here, it was like a kicking task. I think it's from some martial arts or something like that. So people like reached some kind of plateau, but then they were offered to watch some videos how to perform kicks, and then you're mastering a skill, and then you can watch how people do it, notice some details, and then you can improve again. So it also was like shown that observation can.
What is the kicking task? I'm sorry.
Kicking task, it's like participants had to kick. It's like in martial arts, you know.
Kick.
Pinok.
Okay. No ball or something. Just kick someone else. Or?
I can show you. Yes.
So does this refer to all kinds of skills? I mean, we've got motor skills, but then also like linguistic skills. It seems pretty broad.
Yeah, this study was only about motor skills with kicking. I'm not sure about linguistic ones, whether observational learning can help, but I think so. Because maybe when you, I don't know how you can observe like language, you can watch some videos, right, and see and learn from how native speakers talk. But I think it's a bit different because you also learn vocabulary and like, I think cognitive skills are more, how to say, complex, and we will focus more on motor skills. I just wanted before highlight that like we can divide skills in a cognitive skills and motor skills. But in general, if you wanna, for example, learn how to pronounce particular sounds, you can watch some videos how people like move their tongue, and like when you learn French or German with different R's or something like that. I think it can really boost your performance, but I didn't check studies, so I just cannot say something scientific now. Yeah. I'm
Gotcha.
sorry.
Makes
Did I answer somehow? Or
Yeah, I was just surprised there was a reading task and a kicking task and that those were combined.
yeah, well, it was these two tasks were in different publications. It's just adapted images that I grabbed from one study.
Gotcha.
Yeah. So
Okay.
it's not a paper that like used reading and kicking. It would be weird. And they are cited here, so you can look at the original. I just check original and graphs. Since it's old papers, sometimes you look at them, it's like, it's ugly. And that's why I included more beautiful pictures. Well, let's discuss implicit learning a little bit. And implicit learning is implicit, so learning skills without conscious awareness of the learning process. And it's also like a particular feature of skill learning that you don't need to be present. It's not necessary. Of course, it can boost your learning, but sometimes it happens even without conscious attention. For example, one experiment on humans was done. So it's called serial reaction time task. And the idea of this task is that participants had to press a button, depending on which button, which letter was like highlighted on the screen. So really easy. If it's A, you press A. If it's B, you press B, but you should do it really fast. And in order to react fast, you should pay attention, right? But in this task, sequences were were random at the beginning. Then in the middle of the task, for some period of time, sequences were not random. For example, they presented A, B, D, C, A, and again A, B, C, A, B, D, C, A. So it was repeating, and even without noticing, it was not so obvious sequence, of course, not like I told now. They like implemented a sequence that were not super obvious, so participants could not like consciously notice it, at least from a verbal report after the task. But the sequence were there. And they noticed that during completely random bouts of experiments, performance was lower than during these kind of sequence bouts. So participants they not implicitly, they not explicitly learn that, they learned that implicitly. So they didn't notice the sequence, but anyway, their performance increased. And yeah, that's implicit skills. And also another good example, I think it's a bit easier to explain. Everybody probably know HM. I think he's Henry Morrison, right? Guy with hippocampus surgery. So he didn't remember his past, so he had undergrad amnesia. Yes, and but what he could do, he could acquire motor skills. For example, he was asked to trace a star, like it was like 2-line star. Anyway, he couldn't see his arm, and he had to using a mirror draw this star. And he was practicing, and like here is his performance. And then like after, when he was asked to repeat that, he was like, no, how can I do that? I never did that before. Like, it's so difficult. Like, don't ask me to do such bullshit. But then he could do it better and better over and over again, even though he didn't remember that he learned that skill. So it's not necessary to remember that you learned something or not. You don't need that conscious feedback of this process. You just remember how to do that, and you don't need to think about this. Yeah.
You want to say like it's important for motor skills.
Yes,
specifically
for
for motor skills, you can really acquire them without being like conscious of what you're actually doing.
Yep. Yes.
Can I ask?
Yep.
Sorry. So does it also apply to motor skills already acquired before, in this case, the hippocampal surgery, or it only applied, or if they only tested on what he could acquire afterwards?
I mean, I didn't check studies, but from lectures that I heard before, yes. And also, almost all our life consists of tiny skills like brushing teeth, going to the toilet, whatever. And he could do everything. He could walk. He could navigate. Not like navigate in the sense of learning some new navigation, but he remembered past. So yes, without hippocampus, you can, and you remember previous skills, because we will see now that structures that involved in skills memory they're a bit different and they usually bypass hippocampus. Did I answer?
Yes, thank you.
Okay. So we will now look at some structures, because for me it also was a nice recap to see what's involved in motor learning. So main players are cerebellum, basal ganglia, and motor cortex. Thalamus is also involved because basal ganglia usually project to thalamus and then they project to motor cortex. And brainstem and spinal cord, they kind of involved in more basic functions in motor performance. I wouldn't say that brainstem and spinal cord involved in motor learning, but they are necessary in order to perform actions, some muscles control and some balancing out things. And yeah, we will discuss mainly basal ganglia, motor cortex, and a little bit of cerebellum, because I'm not a big fan of cerebellum. And we'll start with basal ganglia. It's always confusing because they're such a complex guys. And I think even though this picture is ugly, you can see better how they arranged with each other. So we have a striatum. It's a really popular basal ganglion that consists from two parts. It's caudate nucleus because it looks like a tail, and caudate, I think from Latin, it's like a tail, and putamen, and together they're called striatum. Then we have globus pallidus, which has two parts, lateral and medial, and then some other players like subthalamic nucleus, substantia nigra, and usually they are playing around with thalamus, but thalamus is not considered as a basal ganglia. Yes, and subthalamic nucleus as well. And what we know about them, we know a lot, and we will see also from clinical point of view, not clinical point of view, what they're doing. But in general, to sum up that, they're involved in habit formation and reward-based learning, and also they help to select appropriate motor actions. And they're involved in refining motor sequences over time. And also they undergo plasticity processes, mainly connected with dendritic trees. So since over time our skills become more refined, their connectivity also become more refined because that's how it works. So we'll consider direct and indirect pathway briefly, because I will not go into details. It's too much. And anyway, if you go deeper and deeper, as I did during my master, it's becoming insane. So in general, we have direct and indirect pathways. And direct pathway is a go pathway. How it works? It disinhibit thalamus. So thalamus here, you can see it projects to motor cortex in order to initiate some action, because we will see how cortex works as well. So when we disinhibit thalamus, we kind of make a system go. So you can execute action in general. And indirect pathway works other way around. It inhibits thalamus, and then and then thalamus cannot excite cortex, and action is not initiated. And these two sides are important because they balance each other out. Especially like at the beginning of learning some new skills, you have to put some effort in order to initiate action. But then you want to refine your skill, your motor performance, and remove some unnecessary extra movements. And here, indirect pathway can go into the scene and kind of make your movements more precise. And we will see now how we can separate hippocampus from basal ganglia. There are some studies in rodents, and first one is about differential effects of fornix and caudate nucleus lesions on two radial maze tasks. So basically, first two papers that we will look at, they were using lesions. I will show you later how lesion look like, especially in fornix. Even though they call it fornix lesion, maybe I will show you now so you understand. It's a fornix lesion. So you basically see it's a big hole in the brain. There is no hippocampus anymore. So we can say that it's actually hippocampus lesion. In subsequent publications that used that all publications, they all call it like hippocampus lesion. So we will stick to that, even though it's like fornix and hippocampus, they are close to each other. So what mice were doing here? Actually, it was rats. I think, I don't know if you know, 8-arm maze. So here, it's kind of working memory task, I would say, but we will discuss working memory next time. What rat should do? Rat should collect food pellets from all arms, but in order to do that, it should not come back to arm that it already visited. So you have food in all arms, and you start somewhere, then you collect a full pellet, then you go in the center again, then you go to another arm, etc., etc. And then the scientists over like trials, they look how many times rat come back and the same arm that it visited before, because rat can understand that if I visited that arm, probably there is no food anymore because I collected that already. So like perfect performance would be if rat would visit arms without repeating visiting it, because it doesn't make sense. There is no food anymore. And what they saw, that rats with hippocampus lesion, their performance over trial did not improve. So errors stayed more or less in the same level. I don't know, at least in comparison to control. And even at the beginning, it was difficult for them to navigate, and because you should remember which arms you visited already and kind of pay attention on some spatial cues as well, because it's sometimes difficult to navigate. But there are different strategies. If we go deeper, we can say like, okay, a rat could like do it like this, right? Like one by one. But the paper didn't discuss that, and I will not do that as well. But with basal ganglia lesion and in a control rats, there were no impairment, more or less, in this test. And then they modified it a little bit. And this time, rat should visit arms in order to collect food. Arms that were enlightened. In paper, they say like enlightened arms. So probably they put some light in that arms. I don't know how. But anyway, food was only in arms with light. And in this case, interestingly, basal ganglia lesion rats could not perform well. Here it's a bit different graph. It's not error. It's percentage of illuminated arms chosen. A bit different. But anyway, we can see that hippocampus lesion rats and control rats perform well, but basal ganglia lesion did not allow rats to perform this task. And so we can see that impairment of basal ganglia lead to difficulty with simple perceptual motor task.
But can you explain this a little bit more? Because this is kind of interesting. So there are two questions, like why is the hippocampal lesion and control differ so much? Do you remember like why? So the control starts, yeah, so the trial one control starts really low. So hippocampal lesion is actually great. Huh?
Actually, you know what? In Bordeaux, people also showed us similar task. And indeed, they showed that when hippocampus is lesioned, it's easy, kind of basal ganglia and hippocampus, they balance out each other in these type of tasks. And when you lesion hippocampus, all your basal ganglia become involved in this kind of stimulus response learning. So you see a stimulus like light and you go. I don't remember why, but it's actually a fact. And I can, maybe for references, I can include some papers. I have the slides left. from the course that we took, but it's true.
So I have an idea that maybe because in general if they put the light into the end of the arm, I mean of course it's less stress for a rat to choose the arm which is light up rather than go. Well, it depends on if it's really dark, completely dark, so you don't know where you're going. Yeah, but if you can see what's at the end, it's a bit less stress, unless it is very bright. Yeah, so they also don't like very bright. So if it's very bright, I really expect the opposite. They would never go to the arm that's really bright. Instead, they just because they don't have a working memory with the hippocampal lesion. Yeah? So in the control is less because they try to remember and they still fail sometimes.
I think in this case, it's not. It doesn't matter whether it's light or some other local cue. It doesn't matter because they also. I didn't show it in this presentation, but there were studies in water maze. We'll discuss water maze, but in different context a little bit. It was like global and local cues, and they also show that when hippocampus is lesioned, you can use local cues. For example, there is a flag, or they put sometimes like some sticker or something on a platform where a rat should swim. And in that case, hippocampus lesion rats overperformed control because they see a cue and perform, see a cue, perform without some extra building of cognitive maps, whatever. For them, it's like stimulus response. So light here.
I mean, so in this case, they learned the task before, so they know that the arm that has a light has a reward, right? So in this case, I just didn't. I don't think you mentioned that here.
So you can see they're learning, so their performance increases.
Yeah. Okay. So
they are naive. They habituated, but didn't. Learn.
So if the trial won and they never been to this maze before, then there's something with the light that they actually attracted by light, right? There's no pattern that you can. Why they are so better than hippocampus? Yeah, exactly. So the controls are really low, like 40%, which is weird.
I don't know if they were trained before. I don't remember.
They also lesioned a little bit of amygdala, so they have they're more brave and they just go to where the light is.
I think so.
I don't know.
Okay.
It's interesting, yeah.
Okay. Do you have guys any questions to this? If no, if yes, okay. So next we'll discuss water maze. It's actually really popular nowadays. People don't use it because it's really annoying. So how it works? Usually it's like a big tank with water, but it's opaque water. It's like not transparent. So when there is a hidden platform, it's actually hidden. So when animal is swimming around, it doesn't see a platform. And there are different modifications. And in this case, scientists also, they lesioned in one group of rats fornix, like all hippocampus actually, and in another group of rats dorsal striatum. And then they used the standard water maze task where animals learn to find a platform, and then they had to remember based on global cues where platform is. Global cues, what do I mean by that? Maybe you don't know. It's some cues on the walls. Usually when it's a real like room, you have some stickers on the walls, even like some posters. People can be creative with that. For us, it doesn't matter. Just they should understand where they are in the space. And what they saw, for example, rats, at the beginning, they swim towards a thin platform. And here in days, you can see that they are swimming to visible platforms, so they can understand and learn where in the space platform was. And then they do probing, so they hide platform, and rats should swim to a platform. At the beginning, performance in all rats are kind of bad. Here they measured latency to reach a platform. So the longer rat is swimming towards a platform, the worse. Because if rat learned where a platform is, it should be straightforward and they should swim towards a platform immediately because they don't like swimming around. And then over days, their performance should improve, right? Because they do it over and over again. And here in day 12, we can see that rats with lesion fornix didn't improve. When the platform is visible, it's fine. But when there is no visible platform, they cannot make sense from the space and they cannot find a platform at the same speed as can control and dorsal striatum lesion rats. So we can say that hippocampus is really important for performing this task in order to navigate. And there are some pictures, like traces, how rats were swimming. So it's a day 12, this probing day at the end. And you can see that rats with lesioned dorsal striatum swim normally, like directly to a platform starting from all quadrants, because in this task, usually when there is a test, a rat is placed in different parts of water maze. So they place it in this place, and it should find a platform in this quadrant, and this quadrant, and here actually you can see it in the plots. But with lesion fornix, their trajectory, like not random, but way worse than in control and dorsal striatum rats. So we can draw a conclusion here that in this type of task where spatial navigation is required, hippocampus is important, but dorsal striatum actually, even without it, it's fine. Then they changed task a bit, and what they did, in this case, rats also learned to swim to a platform, but then the position of platform was shifted, so it was changed. And in this case, rats with dorsal striatum lesions could not perform the task because they, so they cannot, because basal ganglia, they are really important for adapting motor responses based on environmental cues. And with dorsal striatum lesion, rats could not do that. Is it clear?
Well, not completely, because I mean, here what you show is that, sorry, the second is, so this is the dorsal striatum. So the longest latency is the striatum lesion.
Yes, it's an old paper, so it's bad. It's a bit bad, but yes. So it's a latency to reach a platform, and with dorsal striatum lesion, latency is long, so it took for them some time to reach a platform.
Well, it's still a bit strange to me because, like, I mean, you still need to, even to acquire new location, it's not much motor skills. It's more like a hippocampal map. It's like a spatial map, a spatial representation of the environment that you need to relearn. And motor skills are just like swimming, right? So it's...
In this case, we're not talking only about motor skills. We're also talking about that basal ganglia, they're involved in cue-based motor performance. So you see the cue. In this case, like a platform is visible, and you swim towards the platform. And basal ganglia, they involved in that skill where you can see something, you swim towards. So it's a bit like maybe out of topic completely. Maybe it's not a sequence of events, how you understand the motor skill, like jump, then like groom, then you receive a reward. But
okay, yeah, it's a very it's the simplest mode of like goal-directed navigation.
Yes.
Yeah, so like lesion that, then this direct association between the action and the effect is kind of lost. So it's hard to relearn that. Okay.
Yeah, just a skill to swim towards a platform. It's not a, you can see, you can swim. And in terms of hippocampus, you actually build a cognitive map, then you navigate and all that other stuff that builds on cognitive map and navigation. So that's how people try to separate these two systems.
But they still, what's weird to me is like they still swim, as you said, they still swim to the old location. So they have a memory because it's the hippocampus is there. So they have a memory that they have to go there.
Yes.
And kind of the skill is there. And they kind of swim there, but there's nothing there. So I have to relearn.
We cannot say that skill is there.
But they swim there. So the skill is somewhere outside of basal ganglia somehow, like a motor cortex somewhere. So the basal ganglia are very important for actually learning, acquiring of the new skills, not the performance of the skills per se. Seems like.
Can I ask? So they were they trained to like the first platform? Was it learn what it was before the damaging, or then it's not? It's also so it's maybe just okay. So it's preserved that memory, like where it was. It's preserved, so it's not only the task but also the previous memory. Okay.
So it's showing the traces on day 13. And then before they were swimming towards visible platform in upper left quadrant, and then it's showed like disappearing, and they just shifted the platform to a new location. Yeah, okay, from that, let's just remember that skill, in this case, how to say, seeing, doing, it's about basal ganglia, but spatial navigation and all that more complex stuff, it's more about fornix and hippocampus. I don't know if it's too coarse or not, but that's what studies about basal ganglia tells us. Another interesting function of basal ganglia. So here they try to show how basal ganglia activity changes over time in terms of acquiring, like getting better and better in the task. So in this task, rats should go, they hear a tone, they should go and turn left or right depending on a tone. I don't remember, I didn't check how two tones were different from each other in order to go left or right, but it was their cue. So they could differentiate between those two tones probably. So in this case, what they did, they recorded rats with the tetrodes, and on each stage of task performance, like start of the trial, then when tone was presented, turning, deciding on turning left or right, or at the end of the trial, they saw some units that were specifically activated. So they took those events, and shown also here, and then plus minus 1 second, and checked which units were activated in that time window. So they said like, yeah, these units are specifically activated by this event, or at least correlated with this event. And what they showed, probably it's a bit too messy, but if you want data, you can look at this. But in general, here it's more like, how to say, cartoonish representation what they observed. So at the beginning, activation, the most percentage of cells that they recorded were activated during decision turning left or right. But then by the end of the training, late in the training, how they say it, they saw that most of the cells were actually activated at the start and at the end. So, and they concluded that these kind of representations, this association between how units fire in which time of the task, it's changing over time. So maybe it could be a sign that skill is acquired or something, but they show that basal ganglia activity can change with acquiring a skill. Yeah, any questions about that?
So just in case, in this recording, they did move the tetraodes, nothing. So just
No.
record the same area in the basal ganglia, and they got like this transition from one state when it was more about the decision point, and with time it's basically got really connected to like tone presentation, yes, on the tone action.
Actually, I don't know. I cannot lie about moving tetraodes because I didn't look at methods, how exactly they like put a tetrode and how they moved it up and down, or it was in the same location all the time. They just quantified the percentage of all recorded cells, how many cells were activated by tone or by something, and so this proportion were different at the beginning and the end of acquiring a skill. That's all that they are saying in this paper. Yeah.
Mm-hmm.
And they conclude that encoding or control of skills by basal ganglia changes as learning progresses.
Mm-hmm.
That's all that they're telling. Yep. In this beautiful slide, we'll finish with basal ganglia. If you have some questions about them, I'm happy to answer. But yeah, it's a bit complex how they work. And then even more complex, motor cortex. So motor cortex, it's interesting because we know that like there are many animals, right? And not all of them have motor cortex. Like mammals have for sure, and birds have something similar to that, but reptiles, amphibians, and fish does not. But there are some studies on fish that they can do some sequence of events as well. So we could say that is like motor cortex really necessary? And we will see for what motor cortex is actually necessary, for what is not necessary. But what is interesting that mammals are they are more trainable than all other species, because to train a rat to perform a task, it's way, way easier than to train a slug to perform a task or some frog. And that's why maybe we have pets that are mammals more or less, or birds. Yeah. And here you can see I included like pictures because, to be honest, I never checked how motor cortex looks in rodents. And it was interesting to compare. But in general, they also have these kind of Penfield fields where they have like jaw, tongue, eye, vibris, and this one probably everybody saw. Yeah, so it was like interesting to see how it looks like. A little bit of connectivity. In order to understand function of a cortex, we'll discuss a little bit of connectivity, what they found. So cortex receive inputs and outputs. So main inputs to motor cortex comes from thalamus and other cortical areas. So from thalamus to layer 2, 3, sometimes in layer 1, comes input from cerebellum recipient. So cerebellum projects to thalamus, and then thalamus projects to motor cortex. And also sensory cortices project to the same layer. But ganglia-related activity, you probably remember I told you that basal ganglia, they inhibit or excite thalamus, and then thalamus, basal ganglia, they can disinhibit or inhibit thalamus. So yeah, and then thalamus projects to cortex, but in more layers. And frontal cortex also can project to motor cortex. And so our motor cortex integrate all this input, and usually output is the 5th layer. It's bed cells, the big pyramidal cells, and then project downstream, and then all interesting events happen. So what was shown that these thalamic inputs are a bit different, and they play different role. So inputs that go through basal ganglia and thalamus influence movement selection, and the one that goes through cerebellum, it's shaping movement execution. So what we discussed with basal ganglia, what does it mean, influence movement selection? And sometimes it can inhibit or disinhibit thalamus, so you kind of go or not go. And in cerebellum, it's shaping movement execution, because we will see later that cerebellum is important for sequences of events and for timing. So these two pathways and have a bit different input to motor cortex. Yep. So what else? What else? We can also see that there are two pathways, as they said, and thalamic and posterior cortical inputs. They project to superficial layers, and in superficial layers, we have this recurrent activity that modulates motor output. But in case of frontal cortex, it projects closer to a best cells and its direct output. I don't know why this recurrent activity is needed and how motor cortex processes that, but these differences is highlighted in studies that I checked. If somebody more interested in motor execution skills, you can check it out. And yeah, and there are different outputs. Motor cortex project to spinal cord, then between different cortical areas, and also to thalamus to have so thalamus could have a feedback from motor cortex. And let's switch to something more interesting, to actually behavioral paradigms. So first, it was shown that motor cortex is essential for like small fine movements. In this study, it's actually really cute. Rat was had fixed, and it had to perform a sequence of actions in order to get food. And it was fine movements were recorded with many cameras, as you can see. And here, when like sound was delivered, rat had to lift the paw, then supinate, then it's like this. Then hand should be open in order to grab a food, then grab a food, then collect it, so supinate, then put it in the mouth and chew. And they were recording all these events in this rat. And then they used optogenetics, I think. Yeah, let's see. Yeah, they used optogenetics and inhibited motor cortex, right? Right. They inhibited different areas. We can see that they inhibited contralateral sensory cortex, then ipsilateral sensory cortex, visual cortex, frontal cortex, and at the end they show that only contralateral sensory cortex actually showed an effect. An effect was kind of interesting. So here you can see this sequence of motor events in a skill. So lift, hand open, grab, whatever, in this graph. And when they applied like laser and inhibited areas, and here you can see how different areas were inhibited, they saw that for contralateral sensory cortex, when they inhibited it, this sequence of events was postponed until inhibition was removed. So this complex sequence of fine movements were not performed when this area of a motor cortex was inhibited, which is not the case with other cortices. Of course, with visual cortex, who cares? It was probably like a control. Frontal cortex, more or less like a control. But with ipsilateral sensory cortex, it was also not the case. So they concluded that this area, we can see it here, is really important for complex fine movement sequences.
It's interesting that if they inhibit visual cortex, then the taking the palate was way longer. Yeah. So, or I don't know, was the last step of this? If you can go like the chew. So the inhibition of the visual cortex actually affected chewing. So the chewing of the palate was way longer. So it took like, I don't know, 3 seconds, like 2 seconds to chew, 1.5, compared to the control.
The control, yeah.
The A is the control, right?
Mm-hmm. And also bringing this to mouth, like.
Yeah, it's a bit longer, but let's say.
Chewing way longer.
Let's say bringing it to mouth, maybe you need to still, yeah, you already took it, yeah, so you don't need to. I would say like the grabbing should be like longer because you need to see it probably or use the vision. But in this case, the chewing is actually affected, so it's interesting.
But I'm not sure that it's super necessary for them to see it. Maybe necessary, but if it's always in the same place, maybe you can even learn how to where to go.
Yeah. So the initiation of the action was postponed.
Of this kind of complex fine movements action, because we will see later that it's not always the case. Like here. What's interesting, that this motor cortex was shown to be involved in a fine, more complex movements, like in this case, when a rat is grabbing some food, you need really fine movements of your forelimbs in order to do that. But we know also that chickens can walk without head quite for some time. And there are some central pattern generators that can actually take that function, but cortex allows us to perform these complex sequences of events, of fine events. And it's called dexterous movements, if I pronounce it correctly. I'm not native. Dexterous? Yeah. And it was a paper that actually addressed this exact question. So what they did, their point was that motor cortex is important on kind of in the beginning of acquiring some not complex and not fine movements, but then once it's acquired, then it doesn't matter. But motor cortex needed constantly when it's fine movements, as we discussed. So their question was, what's the role of motor cortex then? Because for basic movements, it's at some point does not need it anymore, but for dexterous movements, it's always needed, according to previous studies. And what they did in order to show that motor cortex is not important when it's not fine dexterous movements, they trained a rat to press a lever, then wait for 700 milliseconds, then press again, and then rat could receive water drop. And in these plots, first one, it's a control, and they measured IPI distribution. What is IPI? IPI, it's, how to say it? It's intra-press interval. Yes. So interval between these two presses. And they trained rats to reach these threshold 700 milliseconds. Once it's acquired, rat actually can receive a reward, because when it's less, it's not good. Rat is not receiving a reward. Actually, it's similar with my mice, but they never managed. So yeah, here you can see that at the beginning they are messing up, but then at the end they're going to a plateau and actually perform pretty well at 700. Then what they did, it's trained animals. So you can see that 700 milliseconds already reached. Then they did a 10-day break, and then they make a unilateral motor cortex lesion. So no effect. So rats can still perform a movement, and then they actually lesioned both motor cortex from both sides, and still rats could perform a movement. So once it was acquired, even like bilateral lesions of the motor cortex did not affect this kind of motion, because it does not require like fine movements, right? As we saw before with the rat that grabbing pellet, trying to bring it to the mouth and whatever. but what they did then, they lesioned motor cortex on a phase where rats were acquiring the skill. They didn't reach a plateau yet, and they could not. So that's exactly what they show here, that this motor cortex is important on a level of acquiring skills, but then if it's basic movements, subcortical areas like basal ganglia, cerebellum could take it over, and motor cortex does not need it anymore. It's not needed anymore. So it's also, I think it's interesting because I am not familiar with these type of studies at all, and for me it's like, wow, I didn't know that. Yeah. Oh, disgusting. Yes. So do you have any questions about motor cortex? It's a bit complex, but to sum up, as I said, like fine movements where it's required like some fingers and etc., like we saw with grabbing, motor cortex is constantly required, but in some basic movements, at some point, subcortical areas can take over. And even without them, once you acquire the skill with the motor cortex, then you don't need it anymore. A little bit about cerebellum. I don't like cerebellum, and I put it only in one slide, so we can just go through and then forget about this. But there are some people who really like cerebellum, so maybe they will get offended. But I think among us, there are no cerebellum lovers. So for what cerebellum is important? It's interesting that cerebellum is present in a wide range of animals in comparison to motor cortex, for example. And lesion in cerebellum impair motor skill sequences. So if you need a time sequence of motor events in order to complete an action, cerebellum is needed for that. And it's also important for synchronization with some kind of external anything. For example, in dancing, you synchronize your motions with the music, and for that, cerebellum is really important. What else? It's important for tracking and aiming tasks. For example, there is a basic test on a cerebellum damage. You should close your eyes and try to bring your finger to... Yes, you can try yourself. I'm damaged. Maybe that's why cerebellum is not my best friend. And yeah, and also cerebellum is important for observational learning. When you like dancing and look in the mirror, you can repeat a sequence of events, and cerebellum is really involved in that. And also people noticed and probably studied, I didn't dive into, but I cited a paper that did. They showed that cerebellum has some lateralization of functions. We know in cortex that there is some lateralization. So we have like one hemisphere that a bit differs from another hemisphere. In cerebellum, apparently it's also the case, but I have no clue how it works. That's why we switch to clinical remarks. But if you have question on this stage, you can ask me. I hope you're still here. So, well, let's continue then with clinical aspects, and then we will take a break and paper.
Yeah.
Yeah, good. So, clinics. I think actually in this case, clinical remarks can help us to understand how these different, how to say, layers, structures are involved in motor performance and motor skills. And we will start with basal ganglia because diseases connected with basal ganglia are super popular. And I think everybody heard about that. The first one is a Parkinson's disease. It's when dopaminergic neurons in substantia nigra, I think pars compacta, or reticulata, probably compacta. If I'm not correct, you can correct me, are damaged. And then people have a lot of unpleasant symptoms, like tremor is the most popular one, but also rigid muscles, problems with urination, dizziness, whatever. And there are some treatment approaches like levodopa, but actually it's a bit useless when you lose more and more neurons. Levodopa will not help. So it's really sad. Next one is Huntington. It's a genetic disorder, and it's like how to say, inherited. And there is a mutation in Huntington gene with many repeats of CAG triplet. And when you have many of that, like 40-80 repeats, your Huntington protein is damaged, like it's misfolding, and it accumulates in neurons, and neurons undergo apoptosis, and usually it's in a basal ganglia. And this Huntington disease is also called chorea of Huntington. Chorea in Russian is like chorea Huntington. So what is chorea? It's when it's like a jerky dancing movement and you cannot control it. And also there is a cognitive deficits and emotional changes. So we could draw some conclusions about that, how basal ganglia works, because when they are damaged, you have all of these like chorea, emotional changes. It's actually interesting that there are some emotional changes. Maybe this Huntington protein accumulates in some other areas, or maybe these basal ganglia also involved in something else beyond motor skills. I don't know. Also, Tourette syndrome is connected with basal ganglia function. It's more like polygenetic disease, so there is no exact gene like in a Huntington. So it's like umbrella, more or less, of genes. And it's usually disease connected with a lot of tics, like eye blinking, swallowing, and yeah, that's Tourette. Then it was about basal ganglia, then motor cortex. Usually, what damages in motor cortex could happen? It's like most common is a stroke, and stroke can lead to hemiparesis and hemiplegia. And hemiparesis, it's when it's weakness of one part of the body. It's really common sign of stroke. And hemiplegia, it's like complete paralysis of the side. And when it's like double-sided, then everything is paralyzed. Apraxia, it's interesting because what is praxis? I tried to, when I was studying like many years ago, I tried to define praxis for myself. It's like sequence of events, and in order to complete something, you do something in a sequence. And actually, apraxia is connected with motor cortex damage. So it's inability to perform learned purposeful movements that requires sequence of events. Yeah. Then also spasticity. Spasticity is interesting because with the basal ganglia damage, we have like weakness of the body. So, but with the cortical lesions, we have spasticity. It's vice versa. And probably everybody knows this disease like ALS, amyotrophic lateral sclerosis. I think, I forgot his name, who had that in a wheelchair guy.
Is it Lou Gehrig? Lou Gehrig's disease?
Yes, he and also some scientists.
Oh, Stephen Hawking.
Yeah,
But Hawking did have the LS.
I think yes.
Okay.
Hawking. So it's progressive loss of upper motor neurons, and then there is a spasticity, muscle weakness sometimes, and inability to live normal life. And last but not least, cerebellum damage can cause ataxia, tremor, and dysmetria. Ataxia, it's like a drunk walk. And sometimes people with ataxia actually can be taken for drunk people on the street, and it's not really good disease for socializing, and it could be caused by cerebellum stroke, some tumors, degeneration, but also by multiple sclerosis if it happens, if demyelinization happens in cerebral pathways. Tremor, I actually don't know why tremor is connected to the cerebellum, but apparently it does. And dysmetria, it's an ability to estimate distance to objects. And like I cannot touch my nose because I cannot estimate the distance. Sometimes people with dysmetria also cannot. And this, I think it's a nice time for break. And then we will proceed with the paper and some approaches how to estimate basic motor abilities in rodents. If you have some questions now, you can ask. Otherwise, we will see each other in 5 minutes.
Can I quickly ask also
Yes.
a small question? I think is it also involved in speech, the cerebellum?
I don't know. I think so. But I didn't check it out, so I don't want to lie. Because I think speech is a separate big topic, but I would assume that yes. But I think it's more about cortex, no? Cortex is definitely involved. Everybody would assume that, but I can check it out and answer to you later if you want. That's all that I can say. Any other questions?
I don't know if. Can you hear me?
Yeah.
Sorry, because just my device just went out of battery, so I cannot hear you anymore. But I will just charge it in the pause. Thank you for the answer.
Yeah, sure. Okay. So let's meet in 7 minutes, maybe in 15:20. I think it's nice. See you in 15:20. professional.
Okay.
So the break is over.
Welcome back.
The break is over. About speech and cerebellum, Laura. I checked now during break, and cerebellum is important for articulation and also for time sequence of speech, for its like timing when you say what in a what rhythm, and also for articulation. So if you hear and you've heard me.
Yes. Okay. Thank you. Interesting.
Yeah, and now we will share the screen and we'll discuss a paper that I forgot to send you guys, but I think it's fine. Andrei will present and I will send you later. If you're interested, you can double check this paper. Paper is quite easy, right? So
it's a behavioral paper.
It's
so,
a behavioral paper without recordings. So just about how rats having fun or mice. Can you see something?
Yes.
Cool.
Yes, we can see the paper.
All right, sounds good. So this is a pretty recent paper, although I would expect that they do a bit of neuroscience here, but it's mostly a behavioral paper. So the focus of this study is essentially how the motor performance is affected by a thing called enriched environment. So a little bit of a background. So I'll probably read a couple of phrases. Don't be afraid. I'm not going to read everything, but a couple of phrases are going to be fine, I think, because they put a lot of sense in phrasing it correctly. So essentially, like laboratory animals are often raised in artificial environments with a lack of physical and social stimuli and potentially leading to biased outcomes in behavioral essays. However, in real life, of course, animals live in a more wild and more dangerous, but also more enriched environment. So if you ever saw an animal in the animal facility of a laboratory, which I'm, for example, have to deal on a daily basis, we have here in the Biocentrum the rats, we have mice, we have gerbils. There are other rodent types like ferrets, for example, and they all basically being bred and raised in basically in little cages. So their environment is very is poor, yeah, and at some point people started to understand that whenever we like breed an animals in our artificial facilities, it's maybe not really correct to do experimental work on these animals, because in real life animals have different contexts and they develop different skills. They developed basically their brain is developed in a different way. Yeah, so we also need to kind of understand the consequences of difference between the real life conditions and the laboratory conditions. So again, this nature versus nurture. Yeah, so nurture means a lot. Yeah. So this study is about how the effect of enriched environment can affect motor skills. Yeah, so they've been they mentioned here in this paper there've been a lot of studies how this affects more like cognitive tasks, acquiring the cognitive skills. But here they also want to test what about the motor performance. Yeah? All right, so as a little introduction here, I would do this. So in 1947, so Donald Hebb introduced the concept of this enriched environment, where the rats growing as pets, mostly spending their time outside of cage compared to standard house rats. So they say that basically this cage enrichment leads to enhanced neural plasticity in the hippocampus, improved memory formation, better contextual field conditioning. So this cognitive stuff, is better. So the kind of positive changes in the prefrontal cortex, amygdala, locus coeruleus. So kind of the cognitive stuff, the higher level functions are improved in this enriched environment. However, on the opposite, in the cerebellum, it has been shown that physical enrichment causes long-term metabolic plasticity and increased cytochrome oxidase activity. blah, blah, but this results is a decreased expression of pure neural nets, extracellular matrix proteins. They important for stabilization of neural connections. So it's not like a really positive sign for like cerebellar stuff, for example. So that led these people to kind of the hypothesis that maybe the enriched environment actually improves our higher cognitive functions and abilities, but at the same time, it decreases, as I understood, it decreases the motor performance and motor learning on acquiring of the motor skills. So what they did in the study, they tried to test this hypothesis, and they did it in mice with 5 different experimental paradigms. All right. And the idea of what I'm going to do now, quick within the next 15 minutes, is just let's quickly talk about these 5 paradigms and also what kind of results they acquire. I would maybe right away go to this figure over here. So, I mean, the paper is, despite the fact this is scientific reports, this kind of the very good journal, they're making mistakes. So if you really, at some point, look at this paper, you will find a bit of weird, the weird way, from my point of view, is a bit of a weird way how they structure the figures. And also they make mistakes in the paper, but we'll not focus on that, but focus on the content. So their idea behind this study is that they took 2 groups of mice. It's, I think, 11 and 16, or 12 and 16. One, of course, group is the standard group of mice that were living in the laboratory conditions, like just standard cages, not much to do, standard food consumption, like very standard laboratory environment. And another group that was kind of enriched environment. And then they basically compared these 2 groups across 5 different experimental paradigms that tested the motor performance. So here on the figure 1b, they provide like a rough plot of what each animal had to do. So what is the timeline of their experiments? So on the postnatal age week, 12 to like 16. So this, their main experimental paradigm was the eye blink conditioning. So these are they trained mice on this task. Now, after a little bit of time, they trained on 4 other paradigms. One is the rotarod, another one is balance beam, another one is Erasmus ladder, and another one is grip strength. Although here, down here, they say that they actually do the balance beam, gap strength, grip strength, rotarod, and Erasmus ladder, so in a different way. But I don't know what to trust, but probably it doesn't matter. So this was done for 28 animals, which is quite a big number. So in research, I think 28 animals is a very good number to trust.
Also because it's not implanted.
Yeah, of course, it's just a behavior. Yeah, but still, like recording so much, it's quite some work. So it's a very good number. Now let's go one paradigm at a time. I'll try to explain the way I understood it. You criticize me if I'm wrong or you don't understand something. And yeah, I hope it's going to be interesting. We start with the first out of 5 paradigms. Yeah, the main thing that they claim. This paradigm is called eye blink conditioning. Yeah. So here is on the figure 1a, they essentially describe what it's all about. Yeah, this is the setup when they take the mice, and the mice is on a cylindrical treadmill, so the mice can actually run. It's a head fixed mice. Yeah, so they train the mice to be head fixed and run whenever they want. And the idea behind this paradigm is basically what we discussed last time, is the conditioning paradigm. Yeah? So what they essentially want the mice, the skill to acquire. So they either play a tone or they provide a shine a light. So in this case, they actually decided to shine a light. So there's an LED in front of the mouse. And whenever they shine an LED, after a certain very short interval, like 250 milliseconds, they also induce a nerve path to the eye. So a pretty nasty paradigm. Yeah, so it's not really nice for the mice. The aim of this experiment is after this conditional training. So whenever they shine a light, there's an air puff in 250 milliseconds. Light puff. Yeah, so this is this classical conditioning thing. And what they want to mice to understand that they actually, whenever the LED is on, then even without an air puff, the eyelid of an eye where the air puff is going is going to close. Okay, so first in the very beginning, the eyelid of an eye is not closing from neither from the LED or nor from the air puff because the mice doesn't know. But of course, after some time, just to prevent this air puff, the mice closes the eye. The mice closes the eye. Now, this paradigm can be tracked by this. I hope you see my mouse. You see my mouse? So if you see my mouse, then I'm pointing to this little inset window here. It's kind of the way that they do it. So the light is the conditional stimulus, the air puff is unconditioned stimulus. And before, so what they show is like what was in the very beginning, before the training, then during the training, and then after the acquisition of the skill. So if we look on the how open is the eye at the moment when they shine the light, so the eye is just open, you know, like open here. But then whenever they do the puff, after the puff, there's the reflex, you know, to close the eye. So the eye is kind of closed for a little bit of time and then open again. So this is there's no training. Then during the training, there is this, I would say, association. Can I say that? Okay, well, let's say association between the LED and the air puff that's going to come is start to grow, you know, and what they actually want is that whenever LED is on, then even before the puff, to kind of prevent this unpleasant feeling, the eyelid is being closed. And this motor skill is acquired independent of whether there's air puff or not. Yeah, so in this case, we don't have an air puff itself, but just an LED triggers. So a classical conditioning thing, and what they want to do at the end is just to train both groups of mice and see whether there are changes in how fast they acquire the skill or how good they acquire the skill.
It's actually interesting, right? Because can we actually call it a skill? Because we discussed conditioning last time, remember?
Yeah.
And skill for me, like conditioning for me, is it a skill?
I'm sorry for maybe using the wrong terminology. I don't want to call it a skill. They call it motor performance. So what the idea is to estimate motor performance differences depending on cage enrichment.
In this case, motor performance is the closing eyelid.
Exactly. So in this case, this is the thing that they want to measure. Now, I didn't say exactly, but I wanted to mention it again. Sorry for that. I specifically highlighted it here because for me it was important to understand what is the fucking difference between these two groups. Yeah. So because what do they call by this enriched environment? Yeah. And then I just want to quickly read this couple of phrases. So the standard house mice, yeah, which is N11, but here is N12. So I don't know. One mice, they lose one. So here, this is the N12. But anyway, I'm sorry. Stop that. Yeah, here. So one mouse is lost during the experiments. They housed individually. This is the first difference. So there was one mouse alone, yeah, in each cage. And it was just a bedding and nesting material, and the cage has a small size. It's basically 13 by 13, 13. It's a very small cage. Even our mice here in the biocentrum, they live in the better conditions. I know these cages, we have an example, but it's a very small cage. So usually mice, I think they even like have a bigger.
I think in Russia we had the same.
Maybe. So enriched house mice, N16, socially housed, so they are social, like 3 to 5 litter mates, which is a very big change in the social development. The cage was larger, although not huge, but larger. And they were daily handled by the experimental, although not very much, 15 minutes. But what is the most important, I think, for the motor skills and in general motor development is that they have running wheels, they have climbing rods, they have walking bridges, tubes, and shelter places, sticks to chew, and so on. So which is a very big difference in terms of, I think, motor development in general. All right. Now coming back to this eye blink conditioning, these are the results. Let's go quick.
Maybe you should.
How to do that? Sorry.
Let's find it.
No. Yeah.
You need
Yeah.
this.
Yeah. Amazing. Amazing. So one parameter that they measured between these two groups is the normalized eyelid closure. So what is this normalized? As I try to understand, I need to say that I didn't pay like super deep attention to their methods. I kind of went through them. It was not really clear, but as I understand, this is like how much in all the trials, how much did the eyelid closed? So there were probably trials when it didn't close. Sometimes they close incompletely.
Like misses, maybe?
Like misses or something, yeah. So then it's never like one, yeah, across all the trials, because sometimes probably didn't close. But what they want to show here is the progression across days, yeah. And so here is the group, the standard group. Here is the enriched group, and what they show that on day one, which is very funny. So actually on day one, the response is lower. If you look at this, day 1, day 2, then day 1, day 4, day 7, and 10. Day 1, the mice is actually opening their eyes even more. I don't know why, but whatever. So you see, like it goes down a little bit. Really funny. So the mouse expects kind of likes the puff. But apparently, with the days of training, there is this motor performance. Yeah, it starts to learn, it starts to grow. So by day 10, it achieves some like 0.53% or something.
So blue, it's normal, and orange, it's enriched.
Yeah, orange is enriched.
Okay.
So what we can compare is that an acquisition of this skill is actually diminished. Yeah? So here is like 0.39 or something, which also doesn't match. If I put it here, it's definitely not 39, but some smaller value. But they claim for 39. Anyway, the idea behind this is that the actual cage enrichment leads to a slower acquisition of this eye blink conditioning paradigm. All right. It's interesting that what they did is that for the first 10 days, they trained with a short inter-stimulus interval, which is 250 milliseconds. And then after day 10, they actually switched it to 500 milliseconds, which requires a bit longer, like a bit more complicated to acquire this some working memory is probably involved to maintain this conditioned stimulus in mind to prevent the air puff to reach the eye a bit later. So apparently the next 10 days, with the increase of the stimulus, we can see that the results for both groups, they already knew that the stimulus is going to appear, that the air puff is going to be at roughly like 250. So they start a bit earlier already here. However, now they prolong the closure of the eye and they do it again gradually. By day 20, they already understood it's going to be later. They still try to close the eye, but they keep it closed for longer, which is in comparison to the enriched group is kind of the same effect but slower. Yeah, so the percentage of time that the eyelid was really like closed is less. It's really hard to interpret these results to me, to be honest. Like what stands in between this enrichment, which is like a behavioral thing, and the actual motor performance in this task. So I really don't know if it's if the cage enrichment does the bad or good job, because the test is very specific. And
yeah, it's interesting whether it's a problem with this exact eyelid closure, or it's a problem with conditioning, somehow conditioning learning, this simple type of learning affected by enrichment, and maybe since they're super, like, how to say, advanced animals, they expect some more complex environment, then
Exactly.
they don't see that, they don't pay attention on that direct relations like stimulus response, as we saw with lesions in hippocampus.
Exactly, yeah. So the lesion to the fornix, can actually lead to a faster, a better performance in the early stage of the Morris water maze task, stuff like this, yeah. So which is really strange, yeah, comparing to controls, compared to normal animals.
It's like stupid learning.
Stupid learning, exactly. So maybe this is actually not a positive consequence of standard versus enriched cage conditions. But this is the results, yeah. And they then kind of the next plots is just to consider all the trials, and they just plot it differently. So they and only when there's it's called this not correct response, but conditioned response. So like the when the eye is actually closed according to this presented conditioned stimulus, they call it a conditioned response. So apparently it's like not all the time there's a conditioned response, but they want to separate it by all trials or only when the conditioned response was there, and the percentage of this conditioned response. Basically, all these plots just demonstrate that one group, so which is the standard group, acquires this skill, this association, faster than the enriched group. But of course, we don't know why. Yeah, so it doesn't mean that it's better. So it's just more trivial, maybe, for them to associate stimulus to a particular motor response. And in the enriched group, they kind of, I don't know, think more about should I actually do it or whatever. I don't know. Speculation, a lot of speculation. But these are the results. And this kind of can, in a very naive approach, can demonstrate that oh my God, the cage enrichment is actually not always good, but can be bad. Yeah, can be bad because some of the skills are.
Ask something?
Of course, yes, please.
Did they do any kind of emotional testing? Because it could also be that they are just more reactive because they are more kind of stressed in a small cage, or I don't know. And that's why they are kind of more overreacting towards this buff. I don't know. It's just
Yeah, to be honest, I very agree with you because when the rodent is alone, yeah, it's very stressed. There have been studies that they show that the stress levels are way higher if the animal is growing, alone. So for sure, in this paper, I didn't find any additional tests before. So here is they provide a little bit of, if I can, like, yeah. Standard conditions ad libitum, like experiments in the light phase, 12 hours like direct cycle. Nothing like no performance or emotional or stress testing before that to kind of quantify the differences. I don't know.
Yeah, they could do some behavioral tests like
They do 5.
yeah, but do they? I didn't check exactly. Do they have like a beam walking where it becoming narrower? Because most stressed animals walk differently in an elevated beam. Like we will see.
So we will see. So I mean, for the sake of time, I'm just gonna skip that because I actually didn't really understand the results. It's really weird. But I want to focus a bit more on the rest of the paradigms, and I think describe them a little bit more, because here we're discussing experimental paradigms. There are 4 more coming. So another test that they want to compare these 2 groups is called the balance beam test. What they essentially what they test for is that you have a certain space in the arena where they put a mouse, and then there is this bar that leads to the home cage. And they do 3 days. So 2 days they do a training of the mouse, and then 1 day they do a testing. And it's just 3 times that they train per day. So they put the mouse, it goes to the home cage. So they put the mouse again, goes to the home cage, and so on. But with the training, they also shrink the size of the beam that goes to the home cage. So it starts with 12 millimeters, which is to me is already like very, very small beam. So of course the animal is afraid of falling off. And 6 is I cannot even imagine they go with 6, but okay, so they probably do it. So it's like 3 times with 12 on the first day, and then on the next day they start with 12, mouse goes back home and then shrink it to 6, and then it does also 2 runs with 6 to the home cage. And sadly, they don't report like how many mice fall off or how often does it do it. They just say, okay, we just do this protocol and so on. And on the third day, they test. They test, I think, with like 12 and then 6, 2 times 12 and 2 times 6 or something. So this test is kind of how fast the mouse can go to the home cage, how much time it takes for the mouse to cross the beam. And if we look at the results on the right panel here, so the test for the 12 millimeter is basically not, doesn't show any difference between the groups. Yeah? Interesting, so they in many of their statistics, they don't put any statistical significance or non-significant or whatever. It's just like plot the results, which is okay, fine. So in this case, they don't even, from this very discrete meanings, I don't think they even measure the time in between. So it's just seconds, it's exact seconds. So there's no difference in 12 millimeters, but there's a slight difference in 6 millimeter, such that the enriched group actually did it faster. For me, it's again, it's an interesting paradigm, but in this particular case, it's very hard to infer any kind of impact on motor performance, because it's really not much about the motor performance, it's more about stress levels.
Exactly, it's
And
contaminated.
like, for me, like, it's just like another correlate with like how stressed is an animal or how fast it can reach the home cage or something like this. So, or how fast can it go into this beam? And
it's also difficult to interpret, right? For example, if animal crosses fast, you can say it crosses fast because it's scared, or it doesn't cross because it's scared to cross. That's why it's like a bit slow or something. It's really, you can interpret as you want, because it's again like that internal state that is really difficult to estimate. And now I told that there is a test where beam is kind of becoming narrower, and then stressed animals with anxiety, they stay in a larger part of the beam and they are afraid to cross a tiny one. But when it depends how they did it, if they place just mouse on a thin, like really thin beam.
I think the mouse, they wait until the mouse goes by itself.
Itself?
Yeah. So there's another platform or something before. So the mouse, it just wants to go to the home cage at some point because it's a stress
So
beam.
it would be interesting to see how much time required in order to mouse actually enter the beam, because here they probably calculated this time from the beginning of the beam walking till the end of the beam walking. But the time to enter the beam would be important in this case, because then we could see how long mice hesitated before jumping.
Yeah, actually, it's
into the beam.
a good question. I am not really sure what is the time to cross the beam.
But yeah.
Is it from the time that they put the mouse into the platform or not? I don't know. We need to check.
And also it's tiny time. So look, 30 seconds.
Yes, but like imagine a mouse like 20 seconds on that rod.
Yeah, also kind of.
It's like too much. I think whenever they start, they do it pretty fast. So it's just to overcome the stress of like, okay, I'm not stressed enough. I need to go. Anyway, next paradigm is this grip strength test. Also very interesting. So this on the left side, we see this device that actually measures the force that's been pulled out of this device. And I think they use this kind of, is specific of every rodent that they don't really like when they've been pulled by their tails. So they try to keep everything like not just being pulled by the tail. So it's probably like a good test to measure like the how strength, how also the measure of what. Yes, it's
for limb strength.
Or is it
And
really a
we-
motor skill? Like, is it a motor skill? I'm not sure, but
It's a motor ability.
there's a kind of ability to keep as much as possible to keep the strength, to keep the bar not being pulled by an experimenter. And they did this several times. They did pull these mice to test their strength. And what is the results? Again, on the right side, you can see that this strength for the standard group was a bit lower than for the enriched group. So they were more like stronger. They wanted to maybe keep grip to these bars a bit longer and so on. However, again, no statistical significance. So essentially, like this, we see that there's a bit of a trend, but it's not really like much different between these environmental groups.
Yeah, but also it's expected, right? Because in enriched cage, they're always like climbing, and they even like maybe have more muscle mass.
Exactly. So this is what is bad in these experimental paradigms, is that we don't know what to account for. So is it just because they had wheels and like other interesting, like these bridges and stuff what they had in the cage just led to like more running in general? So it would be cool if they also quantify, which is actually possible nowadays. I know like a couple labs that in the freely moving conditions, they measure what animals are doing, like trying to classify like how much do they run, how fit they are at the end. So like what is their motor performance on a daily basis just in regular tasks. And then we can link it to, having more muscle strength or something. So now in this case, we don't know. It's just a level of stress, which is not related to motor performance at all. Or is it just like muscle volume, or what is the reason for that?
Yeah, but their question was also not to find the reason, but just to show how enriched versus not enriched environment makes a difference. And there they just show like there is something.
There's a bit of a something, but it's not really significant,
Yeah,
right?
it's not precise question, not precise answer. Yeah.
The last one, or the fourth one, is called the Rotarod experiment. The idea is that there's a certain mechanism where there's a rod on which we can put a mice, and we can rotate this bar on which mice sits with a different speed. So, and basically the metric we use here is how long can a mice hold to not fall off the bar. Yeah, so we increase the speed, and of course for mouse it's really, really hard to like get on, to keep going and running on the bar, and at some point it just falls off. And the speeds are from 4 to 40 rotations per minute. And on the right, we can see that with time, essentially, like both groups perform a bit better, but the enriched group from the very beginning is performing way better. So the only like first 2 days are significant. Interesting because also like interesting at the same time so naive and trivial to me, because like if you put like rotating wheels in the cage, which I saw a lot in our laboratory animals, of course they run a lot, and the more they run on the wheel, of course the more they acquire this skill to also be held on the wheel. So the wheel is tilted, right?
No, wait, you mean running wheel? You run inside?
No. So for mice, they have the wheel that's like a small wheel that's tilted a little bit, and they run on one side.
On the top?
Yeah.
Like on the road?
No. It's not on the road. It's not exactly this. It's not. They're not trained to run on the road, but they're trained to run with keeping a bit of a balance. So of course, these mice that were in enriched environment, they kind of perform better from day one. So. I don't know what to like how essential is to compare these two groups in this experiment and relating this to like better motor skills, better motor performance. So
yeah,
it's kind of obvious.
Yeah, it's better motor performance because they actually had the chance to develop their motor
Better the
abilities.
motor, exactly.
And these 3 tests, they did not invite them. They are standardized, and when I was in Bordeaux, people also showed us some standardized tests to assess motor performance, especially like in a mice with some disease models, and they measure all of that stuff. But with BEAM, they usually use like other explain the
Mhm.
different type of the beam. But this rotor rod and pulling by the tail, yeah, and there are many others more funny even.
Yeah, could be
So
for sure.
it's they just use standardized tests.
Okay. Okay. One more is called the Erasmus ladder.
This I don't know.
Erasmus ladder test. Yeah, also completely new to me. So in this case, so what they wanted to test is, so the experiment is the following. So there are two almost identical boxes. This box here on the left and this here box on the right. These boxes are connected by a certain specific stepwise like ladder. So the idea is that the mouse mouse is placed in one of these boxes and it has to go from one box to another and then from another box to the first one back and do it maybe multiple times. They push the mouse out of the box by an air puff, increasing the airflow. So the mouse at some point wants to go out of the box, and then the only chance is to like cross this ladder to go to another box. This ladder is considered is being built in a certain way where the steps are not even. So such that when the mouse is going from one place to another, the correct or the easy way, or the more correct way from the motor point of view would be just to step on the ones that are a little bit higher than the others. And whenever the mouse is not really stepping onto the right one, it's kind of stepping in between, is being considered a misstep. This is another way. Sorry, I didn't read in detail about this paradigm. What stands behind, this assessment as the good motor performance?
Coordination.
Yeah, some kind of coordination. I don't know which brain area and what they want to test with this. But in general, like coordination, maybe motor coordination. And the results from this is that also like there's not much statistical difference in here, but the little slight trend that the enriched group, which of course had tons of stuff in their cages, of course they perform a bit better. So the number of the correct steps in here like has the average of some 70 correct steps on average, and then the standard group has a bit less. Although these two distributions are not super different, yeah. So which is showing that even without all the beautiful enrichment and ability to train ourselves before, it's still kind of more or less fine. And here also the comparison about the percentage of correct steps per day. The enriched group starts with like a bit better results in the beginning. Sorry for the summary of this is basically down here. And also only day one, is a bit different between the groups, but then the standard group kind of understands the task and also acquires like the good number of correct steps, a little bit less, but still, it's not that bad. As a summary,
should we save money on enrichment and just say, fuck it, mice is fine even without.
But this is of course about the motor performance and specifically about these particular 5 tests. Yeah, and I think from this we can just take over. So like what my take home message would be is that I learned from this paper not what they actually wanted to show. More, I learned these different behavioral motor testing paradigms, which I find quite interesting, and I think they fit today's lecture nice. Yeah, so never had to deal with these paradigms before, which might be like interesting to keep in mind for the future experiments and maybe use them as the pre-experimental training in some of the real neuroscientific paradigms. What do you guys think? Do you roughly understand like these paradigms? Do you like some of them or have any questions about them?
Yeah, you can tell first because then I will
How
tell.
can we go back? Oh, yeah, okay, sounds good.
Yeah, maybe motor learning is not exact topic of everybody here, Neither mine, but it's nice to just keep it in mind. And it's interesting that everywhere, like enriched, had a little bit better trend. So at least it was not opposite, only in eye closure.
In the eye closure, which they put a lot of attention, like 2 figures on that thing.
Yeah.
And then they put the 3 next paradigms just in the same figure. So
yeah. But again, if there is a question whether enrichment is good or bad, I think it's always good to keep animals as close to natural environments as possible, because we want to kind of see how nature works, like how biology works. And of course, it should be like, it should be affordable. So it should be like a balance. Of course, putting like a mouse in a tiny cage alone causing stress is not good. And also it was like, I showed you before, hippocampus studies that thickness of hippocampus is significantly different in animals with enriched environment or not. In terms of motor skills, it seems like they can catch up pretty quick. So mice are not required to go to the gym, and do something in order to be successful. But yeah, it's nice to learn from this paper which paradigms are here. There are more, and I can include them in slides after because I didn't show them, but not so many. And usually, as I said, all these tests on motor skills, they are mostly used in clinical research, where there is models that involve like motor impairment, like Parkinson or something, and they use all type of tests to estimate, to assess strength, coordination, blah, 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 because we saw that there are many diseases, and mostly all of them people try to model in rodents, and they should assess somehow motor performance in rodents. So they use a battery of tests and then draw some conclusions. So we, from this paper, learned a little bit about these tests. And if you have some questions, you're welcome to answer. If not, we will discuss next time working memory, which is interesting topic. And I didn't pay much attention during my studies on working memory, but it's a nice background for future topic, decision making, because working memory will be highly interconnected with that. And yeah, if somebody wants to present motor learning paper that I will send in Friday or Saturday evening, you're welcome to be a volunteer. I know that some people already booked other topics.
Motor learning or working memory?
Working memory.
Working memory. Okay.
Yes, working memory. So who wants to present working memory? If nobody, I will have to do that myself.
I can take it.
So,
Oh, okay. Questions?
no, actually, I was about to also offer it because I think the rest already presented or have already topic assigned, but me. So
But
like I was about to offer it, but
between yourselves, for me, it's fine. Who can present? If you like other topics more or this exact topic, you can negotiate. You have time until Friday.
no, it's okay. It's okay. It's just because I also thought that like everyone else already presented. So because I also joined later, so
No.
I don't mind doing it.
It's only.
But I can also take one in the future.
Okay. Did you see a document with the topics? I think I put it in a Google Drive, and there you can open a Google Doc and see not exactly how our topics will flow, because maybe by the end I will change a little bit, because it will be more about like hippocampus, navigation, and we will dive deeper in that. But there will be more and more topics, of course, because we have more seminars, and probably if you will have power and how to say, inspiration, you can take a second round of topics if you want to read more papers, or I will do that. So there are many you can present. So any comments, questions, or if not,
I have
yeah.
a quick question. So I saw there was like in both groups, there were male and female. Did they comment on any difference between genders?
Yeah, good question. None that I read through. So I mean, they don't show on any figures. Yeah, so there's like no distinction. I don't think they tested in this paper. But yeah, sorry, I didn't pay attention to this particular aspect.
No, maybe they just didn't. So
They
thank you.
probably didn't. Yeah, so yeah, it's only, well, now it becomes actually the trend, yeah, to start to yeah do pay attention. It was for years not really like a trend to make a distinction. Now they actually coming back to that, but I don't think they do it in this paper.
But logically, right? If mice works in similar ways as humans, probably females would be less strong in that grabbing task. But in other aspects, I'm not sure. Like,
I cannot comment on that. I don't know. Like I don't
yeah.
know what who's.
And
but
also,
mice is not humans. Yes, it's important to not draw a parallel.
but
Try not to.
usually male rats are bigger than female rats. It's like male cats bigger than female cats.
In rodents, probably, yeah.
Yeah, you can see that.
I mean, gerbils, I'm working with gerbils, and it's not, yeah, the weight is the regular female would be about 70 grams, maybe by 6 months old, and the male would be 80 grams. Yeah, so they would differ like by, 15% or so.
70 grams?
70 a gerbil female and 80 an average, okay? Like an average by like mice is like about 30, 40.
Because recently, who was doing surgery in our lab? I don't remember, Georgi or Arash. Rat was 700 grams. Like, it was like this big rat, really fat. Yeah.
Okay, we need to let people go. No, let's stop that.
Okay, guys, I'm sorry I can talk about rats a lot, but probably it's enough. So have a nice week. See you. And I will send the paper by Friday evening, hopefully, but I will try my best. Bye-bye.
See you.
Bye. Thank you.